Good morning, children. We have started with our lesson number 17, Introduction to Biotechnology from Science Part 2. And children, in our previous video, we have learnt about tissues, types of tissues, and even we have learnt the various types of animal tissues and plant tissues. In today's video, we shall be learning about biotechnology and applications of biotechnology. So let's begin. First with biotechnology. The art of utilizing living organisms and their products for the production of food, drinks, medicines, or for other benefits to the human race or other animal species is called biotechnology. With the help of this technique, it has become possible to produce plants and animals bearing some new characteristics in addition to their natural one. The techniques of bringing about improvements in living organisms by artificial genetic changes and by hybridization is known as biotechnology. Biotechnology includes the techniques of genetic engineering and tissue culture. Its uses are in the production of cash crops, improvement in varieties of cash crops, increase in abilities of plants to withstand environmental stress, vaccine production, early diagnosis of congenital diseases, organ transplant, cancer research, cartilages, etc. in laboratory. Understood, children? Next is tissue culture. Suppose you want to grow a garden like the one shown here around your home or school. What would you do to achieve that? By which method will you cultivate those seedlings? Children, you must have seen flowers of same variety but of 2 or 3 different colors borne by same plant. How is this possible? Let us learn about a modern technique related to farming and gardening. Growth of cells or tissues in an aseptic and nutrient-rich medium is called tissue culture. Nowadays, a complete organism can be developed from a single cell or from a tissue with the help of tissue culture techniques. Tissue culture. First, a tissue sample is taken from a parent plant. Then, this tissue sample is placed in a agar growth medium, where this medium contains nutrients and auxins. Further, these samples start developing into small tiny plantlets, and then these plantlets are planted to the compost. So, this is the process in plant tissue culture. Are you getting me, children? Let's see a video. Tissue culture is another technology which helps to keep pace with increasing food demands and to provide sufficiently fast and efficient system for crop improvement. Cellular totipotency. The ability of a single plant cell to divide and differentiate into a mature plant if placed in the appropriate environment is called cellular totipotency. Now, what exactly happens? Plant tissue culture is invaluable when traditional plant breeding cannot generate plants with desired traits. The culturing or growing isolated protoplast or cells or tissues or organ on nutrient medium under controlled aseptic conditions to produce a complete plant or plant part is called tissue culture techniques. Understood, children? Good. Haberland in 1902 was the first to demonstrate the totipotency and introduced plant tissue culture. It is essential for us to know some important terms used commonly in tissue culture technology. So, have you understood, children? Good. Next is changes in agricultural management due to biotechnology. Genetically modified crops, that is GM crops, are being produced by introducing changes in DNA of natural crops. Normally, such varieties are not found in nature. Thus, new varieties are produced artificially. Different useful characters are introduced in such varieties. Got it, children? Ability to withstand environmental stress. Some naturally occurring varieties cannot withstand environmental stress like frequently changing temperature, wet and dry famines, changing climate, etc. However, GM crops can grow in any of such adverse conditions. As GM crops are resistant to insect, pest, pathogens, the useful of chemicals like pesticides can be avoided. Due to use of seeds of GM crops, there is an improvement in nutritive value and decrease in loss of crops. In this way, as seeds of a better quality of crops are being produced, farmers all over the world are cultivating the GM crops on a large scale. The area under its cultivation is increasing day by day. High yielding varieties of GM crops like banana, maize, rice, potato, soya bean, tomato, cotton, beet, tobacco, wheat, etc. are available. Understood, children? Now moving ahead, applications of biotechnologies. Nurseries are necessary for various purposes like growing gardens on a large scale. Tissue culture can be used to grow those plants on a large scale which bear flowers, fruits of excellent quality. It is profitable in such cases to produce plantlets with the help of tissue culture techniques. Fully grown plants can be produced in a shorter duration. Plants can be grown on a large scale even if means of pollination or germinating seeds are not available. For example, orchids or pitcher plant do not germinate, but these plants can easily be produced by means of tissue culture techniques. Next, in a bioreactor, cells can be grown in a more nutritive medium and protected from pathogen. Bioreactors are useful for producing plantlets on a very large scale. Large number of seedlings or plantlets can be produced in a short time using minimum resources and materials. Usually, plants produced by tissue culture and genetic modification are disease free and also virus free. Embryos formed by conventional hybridization techniques between two or more varieties may not grow fully for some reason. However, embryos produced by tissue culture technique always complete their growth. Rare and endangered plants can be grown by tissue culture technique and can thus be protected from extinction. Similarly, various parts of seeds of such plants can be preserved by tissue culture and those varieties can be protected. So children, these are the uses of tissue culture and biotechnology in case of plants. Have you all understood, children? Good. So, with this, we have finished our today's topic. I hope you all have understood the concept and I have made my concepts clear. Children, watch the video attentively and then read the lesson. Till then, thank you, students. Take care of yourself and your family. Thank you once again.